We are live from the Cold Spring Harbor Labs, broadcasting from the DNA Learning Center. Thank you for joining us and welcome to today's webinar. My name is Joshua Stein and I will be telling you today about the Gramene Comparative Genomics browser. Before we start though, there's a few business items concerning the interface that you're viewing. If you are having problems with transmission, you can lower the quality of the video. That will help if you're having bandwidth issues. And that's on the, if you mouse over the upper left corner of your screen, a little menu will pop up that will allow you to alter that. Another feature is you can go into full screen mode, I believe with some sort of button on the upper right side. Okay. So I think we're going to switch to my screen now. Okay, my name is Joshua Stein. I work with the laboratory of Doreen Ware, and part of my responsibilities is working on the Grameen project. And I'm going to tell you about one aspect of the Grameen project, which is our comparative genomics browser. And this is actually produced jointly with the Ensemble Genomes project at the EBI. So you can actually gain access to this resource both from www.grameen.org as well as plants.ensemble.org, and you'll pretty much be viewing the same resource. Before I go on, I want to thank all of the community databases. They are the ones that are really contributing the data that enables our resource, and without all of this, they're the ones that are producing the science here. So in a nutshell, Grameen adds value to plant genomes. So we take accessioned genomes from the community, as well as annotations from the community, and other data depending on the genome from their primary sources, and we subject them to additional annotation pipelines, standardized annotation pipelines, to look at repeats and genes, expression. We do secondary annotation of proteins, which I'll get into in a minute. And we do comparative genomics analysis, including whole genome alignments and phylogenetic gene trees and Synteny mapping. And then we provide this information in a number of different browser displays, visualization interfaces, and also provide access programmatically through various means, including the Ensemble API, a RESTful interface, Biomart, and a public MySQL server. So this is the front page of Grameen. This is what you'll initially see when you go to grameen.org. There are actually two. This portal actually accesses two resources. One is the Genomes portal, and the other is Pathways. And there'll be future webinars to discuss the Pathways resource. I'm going to focus on genomes today, and so if you simply click on this icon, that'll take you to the front page of the comparative genomics interface. So this is a typical view. This is the initial menu to access various plant genomes. The most popular ones are shown here, but you can also there's a drop-down list to access other ones. If you go to Ensemble Plants, you'll see a very similar interface. So expanding on this front page, you can actually bring up a menu of all species, and there are 38 in total. And I'll just run through them here. This is what I call the Grameen genome portfolio. So 38 species of plants. And these include 20 monocots, mostly cereal grains, but also we have banana. You can see that Our resource is very rich in the Oryza genus. So we have many, in addition to cultivated species of rice, we have many wild species, wild-related species. We have a very strong resource in wheat and wheat-related genomes. Of course, we have maize and sorghum. In addition, we have 12 core eudicots, as well as 1 basal angiosperm. So these include Arabidopsis and brassicas. We have soybean, as well as some related legumes. We have grapevine, and we have potato and tomato. And then we have 5 lower plants. So I'm going to run through some of the visualization. This is a typical genome browser. Again, what we show is a combination of publicly available information that came from the Genome Project, including annotations, the actual sequence itself. We bring in variation data, and then we provide our own analysis. And I'm just going to show you, this is one particular browser. This is the Arabidopsis browser. Depending on what genome and what resources are available, you may see different kinds of tracks. But for Arabidopsis, for example, this is a typical genome browser. So here it shows the annotation. We have EST and cDNA alignments, various array probes that are available for Arabidopsis, our repeat track, variation data shown here, and then various genome alignments, whole genome alignments, and each of these tracks here represents an alignment to a different species. And one thing to note is when you're looking at this view, you can actually click on one of these tracks, and you'll be able to navigate from this page to the corresponding aligned region in the other species. So it's very useful if you're exploring genomes and you want to look at orthologous regions. You can navigate between genomes very easily. As I mentioned before, we add value to these genomes by doing standardized functional annotation of genes. So this includes running interpro scan, which does structural predictions using various libraries, including PFAM and Signal P and others, and these will point out protein functional domains. And here's an example of a visualization of this. And then also from this information, we assign ontologies, in particular the gene ontology, but we also have other ontologies, including plant ontology and trait ontologies. And this information is provided graphically, for example, here, but also you can get all of this information in tabular form so that you have exact details. For variation data, we have SNPs and structural variation for 9 species. And for some of these, we have quite a bit of data. For example, in maize, we have over 50 million variant alleles. But we also have lots of variation in rice, in barley, sorghum, Arabidopsis, et cetera. So with these data, again, we add value. So we take the structural variation, it gets put into our database, and then we perform variant effect prediction. And this is a method, this is an ensemble method that classifies SNPs and other variants based on what the predicted effect would be on the transcript. And we use sequence ontology terms. So for example, a change in a nucleotide may cause a synonymous mutation. It may be a non-synonymous mutation. It may introduce a stop site. It may affect splicing, or it might just be in an intron or intergenic region. So this is an example view of our variation data. This this is focusing in. So as I showed you before, the variation data can be viewed in the browser as a location view. But then for an individual gene or transcript, you can actually navigate to a particular gene of interest and look at and sort of blow that up and look at the SNPs that are available that affect that gene, and they're color-coded based on the variant effect. So here, for example, these blue ones are in introns. These yellow ones are missense variations. The green ones are synonymous variations, et cetera. The red ones are introduced stop codons. So that's very interesting. The variation data appears in other parts of our browser as well. So for example, I mentioned before that we do whole genome alignments. I'll show you more about that in a minute. But one of the things with the whole genome alignments is that you can actually view the actual sequence as in text form, the actual alignment. And in addition to highlighting things like where the exons are and where the conserved regions are, you can see that the variant alleles with using the same color coding are also highlighted here. So in effect, you can compare genomes and you can compare the variation between different species. Okay, and in addition to this, you can drill down for any SNP. You can drill down to a particular allele, and you can look at all of the individual genotypes for the lines that contributed to this population study. So you can bring up the accession names of the plant, and you can view what their particular allele is at that variant. Okay, moving on. I'm going to tell you about our comparative phylogenomics. This is where things get really fun. We do a variety of analyses with the goal of trying to reconstruct evolutionary histories or allow the user to reconstruct evolutionary histories. And there are two main analyses. One is the whole genome alignment. And these are pairwise alignments, and we employ the last Z chain net method of alignment. And I'll show you a little bit more about this, but this is the type of view that you can get, and as I mentioned before, you can do cross-species browsing. The other analysis is phylogenetic gene trees. We actually apply 5 different tree building methods, and these get combined into a single consensus tree. And following that, the method infers orthologs and paralog relationships with all members in the family. And we perform taxonomic dating of events like duplication events. We also provide information such as synonymous and non-synonymous sites. And as well, this allows an opportunity if you're looking at one gene, you can go anywhere else in the tree to browse to a different gene, different species. Here's an example of a region comparison view. So what we're doing here, in effect, is we are stacking the genome browsers from different species, and we're able to look at all of the features from the individual species, but do it in a comparative fashion. So here we have one species of rice, and this is cultivated rice, and it's been aligned to 1, 2, 3, 4 different wild Ariza species. And we've stacked all of the browsers. The green shows where the DNA alignments are. So you can see places, for example, here where there's a gap in the alignment. And then the blue connects genes these blue lines connect genes that are predicted to be orthologs based on the phylogenetic gene trees. So as you can imagine, these types of views are very useful for looking for and discovering insertions and deletions, inversions, duplications, plus it's very useful for finding assembly artifacts as well as annotation artifacts. I'm going to point out a few things about our tree viewer. This is actually an interactive viewer. When you first land on a gene tree, the red here, this shows your entry point. So we started with an Arabidopsis gene. We asked for a gene tree. This showed up. So here is the tree. On the right side is a schematic diagram of the multiple genome alignment, multiple protein sequence alignment. And then you can do various things. So these nodes are actually color-coded. A blue node is a speciation node. A red node is a duplication node. And you can do things like click on a node. A little window here will pop up that gives information about the node. So in this example, it says that this node arose in the Poaceae, so in the common ancestor of grasses. It's a duplication node, and there's other information as well. And if you click on the node, you can expand that node. So here it's fully expanded. You can collapse the node again if you like. You can click down here, and it'll expand to view just the paralogs of the gene you're interested in. So this is an Arabidopsis gene. All of these blue genes here are other Arabidopsis genes that are paralogs. And of course, you could just expand the entire tree. So here's an example use case. This is an interesting example of a domestication gene in the cereals. The shattering 1 locus controls seed dispersal, and in the wild type, seeds dehiss and are scattered, and that's what the plant wants to do, wants to scatter its seed. However, that was not advantageous to early farmers 10,000 years ago who were domesticating crops, and they selected for non-shattering variants. And one of those variants is the shattering 1 locus. In Asian rice, Oryza sativa, there's a 4 kb insertion in the SH1 gene, which limits its expression compared to the progenitor. In sorghum, this gene is also disrupted, the orthologous gene. And so the question here, there's a different species. Many people may not know this, but there, in addition to Asian rice, there's African rice, which is a completely different independent domestication of a cultivated rice that was only 5,000 years ago. And the question, so African rice is, the scientific name is Oryza glaberrima. The wild progenitor of Oryza of glaberrima is Oryza barthii. And when you view the gene tree, this is the Asian rice version here. And you can see that there are orthologs for most of the different Oryza species. We can see the progenitor species, barthii, but when we look for Oryza glaberrima, we do not find it in this tree. So that suggests that, well, either it's missing from the genome or it wasn't annotated properly, or who knows. So to confirm this, we can go to the regional view that that stacks these browsers. So this is the Asian rice in the middle, and that's compared to African rice on top, and compared to the African rice progenitor, barthii, on the bottom. And you can see this is the gene, the SH1 gene in rice, and there's a nice ortholog here in the progenitor. But then when you look at African rice, you can see that there's this whole region here. It's not just the shattering 1 locus, but a fairly large region with multiple genes that's deleted in glaberrima. And this has been confirmed in scientific, with experimental data showing this. Here's another application. As I mentioned before, we have an extensive collection of wheat and triticale, wheat and related genomes, and we have the hexaploid bread wheat genome. In addition to that, we have its diploid progenitors. So the hexaploid has 3 genomes, a DD genome, an AA genome, and a BB genome. And we have diploid progenitors for 2 of these genomes. One is Aegilops tauschii, and the other is Triticum uartu. And you can see here in this tree very nicely that the D genome of hexaploid wheat is closely related to the DD genome Aegilops tauschii. The AA genome of hexaploid wheat is aligning nicely to the Triticum urartu genome. And then we have BB genome. So you can easily reconstruct the history here. And then in addition, there's barley down here and brachydipodium. So this tree looks very, very nice and neat. You can also align the different hexaploid genomes to one another in the same type of view. So you can view the BB genome, DD genome, and AA genome together in an alignment, and you can browse through the genome that way, and you can go back and forth between genomes. Another nice thing, and it's always fun to just kind of explore and view things. This is something I came across this morning. This is an example of a gene family species-specific expansion in soybean. So all of these blue colored genes plus the red are soybean genes, and it turns out that there are 15 copies of this receptor protein kinase in soy. There are only 3 copies in Medicago. Poplar has also expanded, so there are 4 copies in poplar and only 1 copy in peach. So, and there are many examples of this that you can find. Another application, and this is extremely important, we all know that genome data is, reference genomes are not complete. There are gaps, there are problems. Annotation is not perfect by any means. So viewing gene trees and alignment data allow you to spot possible misannotations. So here's an example of a tree, and what we see here, these nodes are actually labeled pink, and the pink node denotes the possibility that this is a misannotation that's called a split gene model. So it's represented as 4 genes in the annotation, but in fact it really should be modeled as a single gene. And you can see that in the multiple sequence alignment, these regions actually don't align. And then you can look at again at the alignment view, the region comparison view, and you can see these are the 4 genes here in banana, and they align nicely to a single gene in rice. So that's further suggesting that these individual exons or these individual genes should actually comprise a single gene. So I'm not picking on any particular genome. We actually see this in all of our genomes, and we provide a list of possible, we call them putative, they're not confirmed, but putative misannotations, and we provide these lists in the FTP site if you're interested. Another way of browsing genomes is to use our Synteny map viewer. And so we construct Synteny maps between relevant genomes. And one nice application is to view homologous regions that arose during ancient polyploidy events. So, for example, we know that grapevine is an ancient hexaploid, and most dicots are derived from that, but many, such as poplar and Arabidopsis, have had their own independent whole genome duplications. And so here, using a common reference of grapevine, one can look at where poplar is synthetic, and you can actually see where there are duplicated regions. And so you can very easily navigate from one duplicated region of poplar into another duplicated region of poplar. The same concept holds true for maize, which is an ancient polyploid, and you can navigate its whole genome duplication using sorghum as a reference. Okay, here's just a few practical points on navigating the Ensembl browsers. When you click on a species, you're taken to basically a species homepage, which just gives useful information about what the underlying data is, where the genome came from, what assembly version it is, what annotation version. You can get more information and statistics, such as chromosome lengths, et cetera, et cetera. You can go to a karyotype view. If you click here, that takes you to the location view. And when you do so, a new tab will pop up that's called location view. And as you browse, eventually you'll end up with 4 or 5 different tabs here at the top. And each of those tabs is sort of a different interface for viewing the data. So here is the location view. So this is the standard browser view, but from this you can also get to the region comparison view. You can get to the Synteny browser. You can look at the actual DNA alignments as a text, as I showed before. If you then click on one of these genes, you can navigate to the gene page, the gene viewer. Oh yes, so here, yes, so from here we're still in the location view, and I'm just showing that you can get to the region comparison view. So anyway, clicking on a gene, that'll take you to the gene page. This gives a whole set of other choices from this menu on the left. All of these pages have a menu on the left to look at different views, and depending on what page you're on, whether you're on a gene page or a location page, it'll have different choices. So here from the gene page, for example, you can look at the Go ontology. And this is in a graphical form. You can also get a tabular form. You can also look from here, you can get to the gene trees, as well as you can get lists of the actual orthologs and paralogs and various other views. If you click on a gene, you can also select its transcript, and that'll take you to a transcript page. And from here, this is the page that you want to go to if you're interested in looking at protein structural domains. So here is on this menu item, it's called Protein Summary. You click on that, you can get a graphical view of the different domains on the protein that's encoded by a particular transcript. Also from this page, you can get to the variation view that I showed before. Finally, if you're on the variation page, you could then click on an individual variant, and that'll take you to a variant page view. And there's a number of menus that are specific to this particular variant, including if you want to ask what are the individual genotypes for the accessions that contributed to this population study or structured study, and other information. Okay, in addition to all of the viewing and navigating that I showed, Grameen, the Ensembl browser in Grameen, is very good for exporting images as well as the data. So you'll never, once you find something interesting, if you want to capture that information, you click on this little icon here, and you can get images in various formats at various levels of resolution, so including PNG and SVG, PDF. You can also click on GFF, and a GFF text file, a genome feature format file, will download that reports basically all of the information that's in this view, including the gene annotations, where the alignments are. If there are SNPs in the view, it'll report those. So basically, anything that you have brought up in the browser, in the tracks, you can actually get the data for where it actually is in the genome. So that's extremely useful if you actually want to study what's going on. Other types of data that can be downloaded or accessed. If you're looking at a gene tree, you can actually get a text version of that tree in Newick format. You could get the multiple sequence alignment that gave rise to that tree. And for individual orthologs or paralog relationships, you can get pairwise protein sequence alignments. Okay, the final topic I'm going to cover today is the ability to add custom tracks. So if you have your own data that you've generated and you want to view it in a browser in the context of other annotations that are available in Grameen, there are ways to upload your data. And you'll be able to view it in the browser, and it'll be private to you. It won't be visible to other people. Here's a list of various file formats that can be uploaded that are supported by the browser. Some of the more common ones are the GFF file format and the BED file format. And here are two examples. This is a GFF, and this was just uploaded from my laptop. This is just an example of tDNA insertions in Arabidopsis. Here's a BED file format, which is basically just a simple form of GFF. And this is showing methylome data. But what's interesting about this example is that it was uploaded from a URL. So rather than physically uploading a file from your desktop, all you do is copy and paste a URL from a website into a box and click go, and it uploads the data. And so this brings me to kind of an interesting use case. So a lot of these public projects are kind of converging on one another, and intercompatibility is a really important factor. And so the projects try to work together to make sure that file formats, there are at least conventions are followed that allows intercompatibility. And one example is the iPlant cyber infrastructure. If you haven't heard of iPlant, it's another NSF-funded project that's run out of the University of Arizona and University of Texas, as well as Cold Spring Harbor participation. Its goal is to provide access and democratize access to genomics tools and data. So people who don't have specialized bioinformatics, training, can actually go to a website called the Discovery Environment. They can bring their data in. There's also, all of the standard genomes that are in Grameen are also available in the Discovery Environment. And you can do things like, for example, RNA-seq. You can align your RNA-seq data and do transcriptome analysis. But the point that I want to point out is when you do an RNA-seq alignment, you're aligning to a reference genome that actually was downloaded and imported into iPlant from the Ensembl browser. And so this enables you then to, once you've done an analysis, that the data that you've generated is compatible and can be viewed within Grameen. And iPlant has a very nice feature where any data that you have, you can create a URL for that data, and you can share that URL. And as I mentioned before, you can also use that URL to upload data. And so this is an example. If you have a top hat alignment of RNA-seq data, this is an Arabidopsis example, And this is looking here at the web iPlant discovery environment, and this is just looking at data. This is the results of a top hat alignment, and one can just click this little box here and then click down here that says send to Genome browser, and it will generate for you a URL here, and all you have to do is copy that URL, go back to the Grameen browser. This is Arabidopsis, and if you click add your data, that'll take you to this interface. You give it a name, you tell it what reference genome you're using from this drop-down menu. You tell it that this is a BAM formatted file, and you give it the URL. You paste it in, and you click attach, and whammo, the RNA-seq data aligned to Arabidopsis shows up on the browser. And not only that, but this is just the simple browser view, but if you look at the comparative browser view, it shows up there as well. So this is very powerful if you have your own data, or if you want to share data with other people, you can send them the URL as well, and do some interesting analyses this way. So this is, again, just summarizing the kind of intercompatibility between Gramene, ensemble plants, and iPlant that allows information to be passed in a very synergistic way. Finally, I'm going to close my talk here. I want to thank again all of the data generators out there, all of the publicly funded projects from across this land and that have made Grameen possible. This is a more detailed list, and I want to thank particular personnel within the Grameen project. So as I mentioned, Doreen Ware is one of the PIs. Pankaj Jaswal at Oregon State is another co-PI. And we also work with Paul Kersey at the EBI. And then particular people that really do a lot of work on this project include Marcella Monaco, who's our project manager, Sharon Way, and at the EBI, Dan Bolzer.